Sorry, I'm running a little over time, but I do hope to give you enough background for the next lecture. So if you have to drop off to another class, don't worry, we'll record a video for you guys, okay? So you can see on the left side, in the last 20 years, the number of GWAS studies really continue to increase. Because genome sequencing is getting cheaper and cheaper, people really want to know, based on my SNPs, are we going to get some disease or not? Try to make predictions. Unfortunately, a lot of the early studies are getting hits that are not reproducible. Even though there's one study that look at diabetes, another study also look at diabetes. They look at the two studies, the SNPs they identified don't really agree very well. And also in the early days, they didn't really get enough SNPs that are interesting. And so this is a study showing that so every spot is one study. And the x-axis is how many samples do they have in that study cohort. And you can see in the early days, they use small number of samples on the lower left corner here, and they end up just maybe discovering 2 or 3 SNPs. As later studies, more and more people are being used. You can see some of the studies are using 200,000 individuals. Then they can identify probably like 100 SNPs that are associated with height or body weight, index, and also cholesterol levels. So there are some known associations of certain populations are likely to develop cardiovascular disease or hypertension and things like that. So those, we know the trait there is association, but really which SNP is there? You need a more, really more populations or bigger studies to really find those. The biggest problem with GWAS studies, if they are not getting the right thing, is population stratification. For example, if we want to look at some simple case, aging, we say, oh, let's just take all the people who live 100 years or older versus those people who haven't, not that they can't live over 100, just take a normal people versus all the centenarians. And then let's look at their population, let's do this study, and then we will find a lot of SNP differences. And by the way, you're doing like a million chi-square test, right? If there's no population stratification, you look at the p-value distribution of all your SNPs, you should see on a QQ plot that they really lie on the QQ plot with only a few spots on the top, or not on the QQ plot. These are the significant SNP hits that are really associated with some disease or a phenotype. But then sometimes when you do a GWAS study and after that you do a QQ plot of all the p-values on the different SNPs, you might see a p-value distribution like this, which means like almost nothing really fits what you expected. And very often this indicates the populations you selected for the case and control are not really matched. In the aging study, it's possible that just by chance in the centenarian population, it's a lot of Japanese. And I think we know that Japanese, they have a good longevity for whatever reason, maybe because of their diet, not necessarily because of genetics. You happen to selected more Japanese in your case, whereas in the control is just general US population. And so at the end, what you are seeing is the Japanese US is probably more Caucasian population difference. You see all those racial differences, which has nothing to do with aging. And that's when your p-value will no longer be on the QQ plot. There are ways to really adjust for this, to really kind of change, give a scalar to move all the p-values down. And so basically, in order to really detect that, sometimes people don't really know, whether the ratio things, you can't just look at skin color. And so very often after genotyping, you could use all the SNPs and draw a PCA. So remember, SNPs is, like there are a million SNPs, and each person has a genotyping information on the SNPs. So if you look at all the individual versus all the SNPs, it's a very big matrix as well. You could do principal component analysis, and in this study, very quickly you could see in this population, even with 2 principal components, you can tell which ones are people from Africa, which are people from East Asia, Central Asia, or America. These are the native, I guess, American Indians versus the European populations. And so you want to make sure that your case and control are really matched for this. Otherwise, if all your cases are the red color and the controls are the green color, then you are not really getting the correct comparisons. Another possibility is in the populations that you are getting, sometimes there are people who are related. They may not know they are related, but they, maybe they all share the same disease. They get selected. They didn't know that their distant cousin also have this disease. And so when you have very close related relatives, that also skew your statistics. For example, this is a parent and their child. If you look at the 2 children's genome, they are very, very similar because, they just got their genome from the parent. And then they each got married, and then they have their children. The cousins also have a lot of genotypes that are similar. So there is a test called identity by descent. They measures across all the alleles, how many of those are the same versus a different, to really evaluate the likelihood that they are related. Actually from this, you can see there are twins that they didn't know they were twins. Maybe they were raised separately. There might be cousins that they didn't know before. And so those, you need to remove those from the study. Otherwise, you kind of have repeated measure of something that's not representative. So you only want, so basically any of those close relatives, you want to remove them. And so basically by this time, there are, this is only the 2012 statistics. NIH has a database called the GWAS Catalog. It has all the recent published GWAS studies and all the SNPs that have a reasonable p-value cutoff. They are using a loser cutoff because they want to have a complete collection. You can download the p-value of each SNP. You can filter later. And each color here indicate, this chromosome at this location has a SNP that's known to be related to some phenotype. And this is the 2012 collection. This is continuing to increase over time. And so we have a lot more information. And once they do this, actually they found most of the disease-related SNPs are on non-coding regions of the genome. If it's on genes, it's like, oh, this gene is related to the disease, you understand it right away. But it turns out most of the GWAS hits are on non-coding regions of the genome. And so they want to understand what's going on. One way to do this is by something called expression quantitative trait loci, or eQTL. And so one project that really allow good eQTL analysis is a project called the GTEx. Basically, they took 1,000 individuals. These are people who probably died from some trauma, car accident or something. And so they take 54 different tissues from these individuals. It's like donors from people who died from accidents. They can take their tissues and then they do DNA, RNA profiling of those individuals. And then you can ask, are there SNPs in those individuals that are associated with expression changes? So in this case, we are not using hair color or the likelihood of getting a disease as your trait. We're using gene expression as your trait. That's why it's called, and also because gene expression is not yes, no, express, and not express. Disease is usually just binary, yes, no, whereas for gene expression is a quantitative trait. And so we are trying to ask what type of SNPs in the genome are associated with the expression level of genes in those hundreds of individuals or 1,000 individuals. And so for that, eQTL analysis is something like this. For example, at the one SNP location, you ask, okay, this is just 2 alleles. How many individuals have one allele versus the other allele? You can also, by the way, have TT, GT, and like heterozygous and homozygous. And then in that case, what is the gene expression level of the homozygous GG, the heterozygous GT, and the homozygous TT? And usually a good analysis, a good SNP hit is that by the way, if there's a heterozygous, hopefully that's right in the middle of the two. That's an indication that when you have a particular SNP, it's related to the nearby gene expression. And people have used this to really annotate a lot of the non-coding SNPs in the genome and say, oh, when this SNP changes, because we have hundreds of individuals, you can really see when it's GG, maybe this gene is low. When it's GT, is higher. When it's TT, is even higher. And it's statistically significant. You can find those links between a non-coding region and the coding region. And that kind of establish what happens with the SNPs is probably regulating the nearby genes. Okay, so we will, so in summary, in the genome, there are a lot of SNPs, and the nearby SNPs are also in linkage disequilibrium, and they form these haplotypes, blocks. For GWAS, people either use the family-based association studies to see which of the parent alleles are being transmitted to the affected child, or they use case control studies. This is just, look at, again, each allele, how much it appears in the case versus control. We need to make sure that we pick the right population in the case control studies, that they're really matched. You can increase the statistical power or reproducibility by looking at just haplotype blocks instead of each individual SNP separately. You can also increase your sample size, and also make sure you reduce population stratification and eliminate those patients that are unknown relatives. And now with GTEx, because most of the GWAS SNPs are non-coding, people are trying to use eQTL to link the non-coding SNPs to the nearby genes they potentially regulate. And in the next lecture, we also have a guest lecturer who will teach us about how to use other functional genomics data to annotate these phenotype-related SNPs. Okay, thank you so much for staying with me.